This is a production of Cornell University.
So today I'm going to tell you a story about rice and water. I think that's an appropriate story to tell. So rice and water. So rice is a perennial semi-aquatic plant. It covers 10% of Earth's arable land. And most rice is grown on farms that are less than a hectare in size. That's about 82% of the rice on the planet. And over 90% is consumed within the country where it's produced. And those statistics should tell you quite a bit already about what I'm going to situate you for, which is if most rice is grown and consumed locally and not traded internationally, that leads to much greater diversity in terms of the landscape. Rice production in terms of countries, the three largest countries in the world account for over 50% of rice produced. That's China, India, and Indonesia. And when you look at this little map showing where across the globe we have rice production, I hope that little red spot comes forward. That's the real reason why China and India outproduce other countries. It's not because of the land mass, it's because those red areas signify that they get 3 seasons per year. So 3 crops on the same land. And you'll notice if you look across, the blue is showing you the areas that produce 2 crops per year, and the green is areas that produce only 1. Now in Asia, which is where the majority of rice is grown, what you see is also that within those countries that are producing the majority of the world's rice, there are a vast number of different varieties. In fact, thousands of varieties are produced every year. And because, as I said, they're produced and consumed locally, every locale in much of the rice growing world has its own favorite rices. And they don't really trade their rices widely. So if you go from village to village, you will be treated to rices and cuisines that have evolved with those rices that are unique to that village. And that leads to preferences that have to do with quality as well as to the biology or the edaphic constraints of the region where they're grown. Now, it's interesting to note that the relationship between production and export is quite unequal. And so what you see here are that there's only 10% of rice that's exported from any country. So it's a very inelastic supply. There's always more demand for importation of rice than there is rice in the export market. But you see that there are 2 countries that predominate. That's India and Thailand, and Vietnam being a close third. And the reason is because there are really only 2 types of rice that are exported in any large quantity. Many of you sitting here will know which those are. They're the 2, what we call supreme quality rices on the international market, Basmati and Thai jasmine. And those 2 rices alone account for over 80% of rice that's exported, because people are looking to pay for high quality rice on the export market, even as countries like India are importing lower quality rice to feed their people. So it's a very interesting kind of market that we have, and it's unlike that of the other major cereals. Wheat or maize, for instance, are normally very big commodities in the international market, and they're not prized so much for their local attributes in terms of cuisine. Now, the other thing about rice that I think is very interesting is that we have these different ecotypes, and a lot of the genetic diversity of rice is partitioned partly because it's an inbreeding species and partly because there are adaptations and whole germ pools, gene pools that are adapted to these different rice ecotypes. So this is about rice and water, so I've divided them into the dry and the wet. And on the dry side, we have the upland rice, which accounts for only about 9% of the land that's used to produce it. The rain-fed lowland rice, which we call dry rice because it's completely dependent on rainfall. So sometimes it's too wet, but often it's too dry. And then there are the wet rices, and you'll see from this topographical map that when you get into the wet rices, these are rices this is that semi-aquatic nature coming forward. You have the irrigated paddy rice, which accounts for about 50% of the land planted to rice. You have the flooded day crew, which is an idea that rice is planted on the shores of rivers, and the rivers flood every year. And also, it's often planted on the shores of the sea. And so these are saline type rices as well. So mangrove swamps and areas that flood periodically. And then you have the deep water rice. And we published a paper about a month or two ago in Science about the mechanism of deep water rice, where rices can rise very, very rapidly to keep up with floods that come and stay for months at a time. But the interesting thing about the talk I'm going to give today is not going to talk at all about the deep water rice, which is grown on only 5% of the land. I'm going to talk more about the problems that we're facing as a community as we think about maintaining rice production into the future. So irrigated paddy accounts for production on about 50% of the acreage that's planted to rice, but it accounts for 75% of the rice that's produced. So it's a very, very productive ecosystem. It requires that you have control of water. So the concept of irrigation is not just that it's standing water, but that you can put water on and take water off at will. Now, normally you do not plant into standing water. You plant into very low paddy soil, and then you bring the water up, and then of course, before you harvest rice, you have to be able to take the water off and dry it down. And the future, part of the future's discussions that are going on in the international community is about the concept of what we call alternate wetting and drying, where you really need control over water. You can bring it in and take it off, and you can actually pull the water off mid-season. And that stimulates a lot of root growth, and it stimulates, in some cases, better productivity, but then you want to bring it in during grain filling. Anyway, so the irrigated paddy is the most productive ecosystem. The rain-fed lowland rice accounts for only 19% of production, but is planted on about 36% of land. So you can see that the ratio is quite different. And the rain-fed lowland rice is actually a major ecosystem in many countries, and all the poorest farmers end up planting on either rain-fed lowland or the upland and the deep water environments where they have no control over water. So control over water in the rice production system means that you have the potential to manage the management is usually more sophisticated, and you're not dependent on rainfall. So now I have to introduce you to at least a couple of these ecosystems so I can go on and explain further. Many people look at the paddy system and they think, what are we going to do? We can't produce any rice in the future. Look at that standing water. We don't have enough water on the planet to continue to produce rice in this way. So I just want to walk back a little bit and explain something that you may already know and some of you may not know. But the paddy system has evolved partly to take advantage of wet land. So there's land all over the world, of course, but a lot of the land in Asia actually is right on the edges of rivers. There are a lot of major rivers that flood every year, and the only crops that can survive in those flooded areas, the only major cereal crop that can survive is rice. And the other real explanation for why people flood and want to continue flooding the land is because it's the oldest form of weed control that we know. So if you have a crop that can grow in standing water, and as I've said, rice is the only major cereal that can do that, then when you bring water up to a certain level, you do two things. You drown out most of the weed species, if they're not closely related to rice, or if they're not an aquatic weed, and you provide standing water, stagnant water, when it goes anaerobic. We'll push the pH to pH 7, and that optimizes the availability of nutrients in the rice paddy. So having flooded rice, standing water, an anaerobic situation, you give rice an advantage over weeds, you give it almost the perfect nutrient supply, and you're using land that could not be used for anything else. And that's sort of the history in a package of why we produce, or why the world has moved towards this kind of production. And I just want to mention that the reason rice can do this is because it's capable of taking oxygen down from the shoot down to the root through the parenchyma cells. But it has to have a shoot above the water line in order to be able to do that, because it has to access oxygen from the air. So it oxygenates its own roots by this system of passing oxygen through. Now, one of the other really big problems with the rice paddy today, although it's optimal for growing rice, is that among the microbial populations in the rhizosphere and in the paddy as a whole, in these anaerobic soils, are the methanogens that are pumping out methane. And that puts a lot of pressure on the rice community to, along with the lack of fresh water, to move out of the irrigated systems. So I just want you to understand that when we think about moving out of the paddy system, we're going to have to deal with things like weeds and nutrient disorders that the rice community has not had to deal with as it's bred rices for these irrigated systems. So you think about the other extreme. If you have control over water and you can irrigate, you're very productive. The other extreme is that you live in these upland areas. This is the absolute poorest part of the rice producing community. This is a slash and burn field in Laos. And there's very poor soils throughout this part of Southeast Asia. And when you slash and burn, you only get about 2 or 3 years of production before you have to move out and leave the land fallow. But the origin of upland rice is quite interesting because these rices have evolved in these environments for thousands of years. And people have learned to make selections. These are usually traditional varieties. These are not professionally bred. And there's a lot of manual weeding and a lot of anxiety about drought. So I show you this picture. These are Laotian community in the slash and burn hills. But there are other parts of the world that grow upland rice in a very different way. And this picture shows you the cerrados of Brazil. So when they cleared parts of the Amazon, they started planting rice in these cerrado areas, which are upland in the sense that they're very dry. And they plant rice with usually they plant it the way we do in the United States, completely mechanized. They've slashed and they burned the Amazon, and they go in and early on, before they started planting pastures, they planted rice, and they planted upland rice, and they planted it with machines, and they left it there, and they let the weeds compete, and they didn't irrigate, and they didn't worry anything about it, and they came back in at the end of the season and harvested, and they got something there. So they started breeding their varieties for this very extreme upland ecosystem. And those varieties today are very, very in demand, especially through parts of West Africa where rice consumption is growing the fastest. Okay, and the only other ecosystem I'm going to talk about is the rain-fed lowland ecosystem. And this one has benefits and some very severe challenges. So what you see in the top left is the droughted area. This is a rain-fed lowland field where it hasn't rained for a while. And this gentleman is actually replanting, trying to fill in areas where his rice is thinning due to the drought. And you will see people going in and trying to replant seedlings in these areas, hoping that the rains are going to come. In the lower right, you see the flash floods, which is the opposite problem. And this is constantly a problem, and it's a growing problem with climate variability, where you have both drought and submergence in the same season. And traditionally, people had a rainy season and a dry season, and they planted very different rices during the rainy and the dry. But now they're getting rain during the dry season and drought during the wet season, and we're having to breed for things that can withstand both drought and flooding in the same season. On the opposite poles, you see problem soils that are in parts, one is in the top right is in Colombia, the lower left is in Liberia, but these problems are very similar, where you have iron toxicity, unavailability of phosphorus and things that go along with acid soil problems. And the breeding in those areas is actually also a very interesting endeavor, and one in which I've been very involved and continue to be involved. This one on the lower left is a picture of the iron toxic fields in Liberia, and you can see a few varieties in the front rows that are actually doing quite well in that soil. But there's been a long history of people looking for tolerance to iron toxic soils in the Glaberiba, which is the rice that was domesticated in that part of the world. Anyway, just to move on, so I'm going to talk a little bit about the geography of these ecosystems and then tell you something about the breeding. So the geographical distribution of these ecosystems, if you just look at that map, you see a bunch of blue dots. Those are the irrigated fields, and they're concentrated throughout the high producing areas of China, India, and Indonesia. those three large producers in the world. You also see some blue dots a little bit down in Uruguay, in South America, in Madagascar, in a couple of, in Mali, there's a few areas in Senegal where they irrigate, and then in the United States in the Mississippi Valley. But what you see throughout most of Africa and parts of South America are blue and red dots, indicating the rain-fed lowland and the rain-fed and the upland, which is of course rain-fed in a sense. So the parts of the world that are producing rice outside of the irrigated system, those are the parts of the world where rice consumption is growing the fastest. And they, as you see, they don't have controlled irrigation. So breeding for rain-fed lowland and all of the incumbent problems that come along with it is actually servicing a great part of the world. You'll also notice that within, in Asia, you see Southeast Asia and vast parts of India and Nepal where you have rain-fed and upland systems. So these are the parts of the world where the breeding has not been very efficient in terms of the Green Revolution. The Green Revolution breeding, those semi-dwarfs that went out, are all require controlled water and controlled fertilizer, pesticide, and usually the beginnings of mechanized labor. So when we align the geography of ecosystems with the climate risk map, of course you see that they're on quite expectedly across these areas where we're talking about rice production being focused in these problem areas with what we call non-ideal growing conditions, meaning they're the rain-fed lowland and the uplands. Those are the areas also that are being exposed to the highest climate risk. So what are the rice production challenges? So, you know, I've worked my whole life really, it's funny to be working on rice and talking about rice breeding here in Ithaca, New York, because most people don't think about it every day, and they're certainly not that familiar with these environments. But for me, this has been my life, and I think a lot about what the production challenges are in the parts of the world where rice is not only a source of food, but it's also a livelihood and a cultural way of life. So the rice production challenges that we face are to accelerate the breeding of high-yielding, climate-resilient, and resource-use-efficient varieties for these different environments. We have to reimagine how rice is managed, and this is a huge endeavor that involves many people, some of whom are in this room, thinking a lot about agronomy and water management, and also thinking about when and where rice is produced. It's interesting that China is already opening up new rice production areas in the northern part of China, north of Beijing, counting on global warming to move their rice production zone from the south farther north as the world, as the climate warms. We're also in the process, and very actively so here at Cornell, of developing predictive models to help optimize the genotype by environment, by management, by microbe, and that's predominantly the methane-producing microbes, to optimize those interactions in future rice production environments. And then to improve the connectivity among breeders, farmers, millers, service providers, input vendors, policymakers, and consumers. And there's a lot of work going on right now. As you know, I wear another hat, which is the director of the Digital Ag Initiative here at Cornell. There's a lot of work going on in digital ag, especially in India and in China, to try to leapfrog over some of the problems that they're encountering with the traditional forms of extension and outreach, to use digital technologies more wisely to try to help make these transitions that are going to be needed to feed their people. Oh, and I did want to say, so I have these themes of genetic diversity, resilience, and resource use efficiency. And today I'm going to give you some examples that are going to interweave some of these ideas as we go forward. So those three themes will be coming back around. So I think it was Jim who said when I graduated from Cornell with my PhD, I moved with my family and my two little kids to the Philippines. And if you're a rice breeder, this is the most beautiful thing you can do, is look outside of your lab and see all the rice paddies right there. You can imagine all the experiments that you might want to do. Those are those electrophoresis units. That was RFLP days. That was Southern analysis. That was in the early 90s. So our RFLP map consisted of 135 RFLPs, and it had taken us 2 years with 3 people working 24/7 in order to produce that map. So you have to kind of think back to what the early technology was like. And as Jim said, I did a lot of training, and during those early years, we not only trained at IRRI, but I actually went out to most of the national programs. And in the early 90s, China was in a very, very, very different place than it is today, and trained many of the people who are today ministers of agriculture and doing wonderful things for their country. And it was back in those days when I don't think any of us could have possibly imagined what that country would do in terms of agriculture. So my time at IRRI was also punctuated by my love of the gene bank and all that it represents. So that's just a picture of a brick and mortar building. The gene bank actually isn't in that building, it's beneath that building. The gene bank sits in an earthquake-proof vault underneath that building, and my lab was in that building. So all I had to do was go downstairs, and there were 180,000 different accessions. And they were just sitting there, these seed packs, in, most of them unused. We didn't know much about them. Usually they had passport information, which is that you know where they were collected, when they were collected. And sometimes you knew a little bit about some of the morphological traits, but you really didn't know much about them. So there was this sort of enticing opportunity to use this stuff, but where to begin when there are 180,000 of them? So one of the things you would do, of course, was think about the ecologies from which they'd been collected. That would tell you something about the genetics. You could think about the geography, and that might tell you something. You could also think about any comments that had been made and registered on those seed packs when they were collected about what farmers used these different rice varieties for. But my point is, and I'll come back to this at the end, is I've been fascinated by the idea that traditional farmers had a kind of closer communion with the land and with the ecosystem and with the challenges than most of us have today. And therefore that there was a lot embedded in these seeds that if we could just extract that information, it would be useful to us as breeders. Now, the other thing is that, of course, when I went to IRRI, I had to decide what to do. And we had a nice map. We had all 135 markers. That was a big deal. But there were a lot of challenges that needed to be addressed. And so the Rockefeller Foundation, who was funding my position at IRRI, had done a study to determine which traits were the major constraints to rice production in the environments in which they were interested. And they had helped to target my work and the work of many others at IRRI. So the next part of this story is about what traits, what discoveries, and what outcomes we have seen over the last, that was in 1990, so if I calculate that, I think that's over 25 years, over that period of time. So one of the first traits of interest was the concept of roots. And roots, of course, have something to do with water and nutrients and anchoring and all kinds of things. But nobody had really got a very good handle on the genetics of roots at that time. And so that was one of those tantalizing opportunities, was to see whether these different root structures, which you see in this photograph, could actually be tracked, were tractable as genetic characteristics of the plant. We also looked at salinity and submergence tolerance and iron toxicity tolerance and a variety of things that had to do with the soils in these rain-fed areas. And I was a pathologist coming out of a pathology background, so I was also very interested in diseases. That first one is rice blast. The one to the side is bacterial blight. And then the green leafhopper is a transmitter of a virus, of the yellow mottle virus. And so these were early on, of the Tundra virus, I'm sorry. These were early on the kind of traits that I was looking at. And I had a relationship with a guy who was a soil scientist there. And through that, I developed an interest in understanding nitrogen fixation in the rhizosphere of rice. And it turned out that this was genetically tractable. And we published the very, very first paper showing that you could track genes in the plant that corresponded to the ability to fix or stimulate nitrogen fixation in the rhizosphere. This is associative nitrogen fixation. This is not nodular. But there was obviously some kind of exudate coming out of those roots. We didn't have the tools at that time to study further either what the exudates were or which microbes were doing the fixation. But I just wanted to mention this paper was published, as you see here, in 1995. And so that was a very early start. And the material that can be used to pursue that today, if I were looking at microbiomes today, that's one of the things I would certainly pursue. I was also fascinated by domestication and, as I said, the early land races. So what does a geneticist and a breeder do? You have to do a lot of crossing. Well, crossing in rice, you see this picture. You have to actually emasculate every single floret that you're going to cross, and it's quite a laborious thing. You clip the floret just before the anthers are up to the top, and then you can make your crosses. It's done. It's still done manually throughout all across IRRI. And we were developing populations in those days, mostly biparental populations that were the source of QTL mapping. So remember, this is 1990, okay? And this was a big deal because this was an opportunity to get at traits genetically that had only been able to be under selection by breeders at the phenotypic level. And so we were looking for parents that were, of course, they had trait contrast. So in one case, susceptible or resistant. They had to be genetically divergent because we only had 135 RFLPs. And you want those RFLPs to be polymorphic. So if you take two lines that are very closely related, you wouldn't have enough markers to cover the genome to do a QTL analysis. So a lot of these were indica by japonica crosses. They had to be crossable. We wanted fast generation time because we were making these immortal recombinant inbred populations. And I won't go on about the details of this, but I just want to say that many of the populations that we made in the early 90s when I first went to IRRI are still the basis of what is being used to derive and clone genes today, and also to drive a lot of the breeding that I'll talk about in a moment. These were indica by tropical japonica crosses. The countries of origin were things like the Co39 by Moribiricon. They were from India and from Cote d'Ivoire. They were irrigated by upland crosses. They segregated for all kinds of pests and diseases. They segregated for root structure. They segregated for drought tolerance. They segregated for seed size, all kinds of things. And so the bottleneck, of course, was phenotyping, because you could genotype one population if you could create the population, and it would take several years, but you had 2 or 3 seasons a year. So you could do this fairly rapidly. So all I'm going to say is we created several populations, and those populations are, in fact, a source of wealth today. So if you can get your hands on them, they're publicly available. And if you're ever working on rice, and I'll tell you more about the kinds of resources we've built, these are really good opportunities. And I will say, I have the seeds right here at Cornell, should anybody want to pursue it. So one of the early papers was this paper called Locating Genes Associated with Root Morphology and Drought Avoidance in Rice via Linkage to Molecular Markers. And my graduate student, Mike Shampoo, who you see up in the upper right-hand corner, led this work. So it was published in 95, and I only got to IRRI in 1990. And it was a lot of work, and I want to just walk through what we did. So we grew rice in the greenhouse using these PVC pipes because we were measuring root depth and doing some calibrations on root thickness. We grew them in aeroponics. We didn't have hydroponics, but we had aeroponics. And if you look at the length of those roots, those are only 6-week-old plants. These were because we had an upland variety in our cross. We also grew them in the field and withheld water, so it's what we call managed stress. So we were able to apply stress when we wanted to because we had irrigation control. And then it was all RFLPs. And so RFLPs, of course, are really cumbersome and really slow. And you can see the Takaishi there grinding the tissue on liquid nitrogen. And we produced this. This is the map. This was the map that I produced here at Cornell before I went there. I think we had about 150 markers in those days. And those little boxes you see to the left of each of the 12 chromosomes are indicating where we had, let's say, QTLs associated with root morphology, either from these greenhouse or aeroponics, and where they overlapped is the coloration or the barbershop poles on the chromosomes where they overlapped with field level drought. So we were starting to show that you could find regions of chromosomes where you could associate root traits and root morphological characteristics with performance under drought in the field. And this was absolutely revolutionary. And when I took this result to my boss at IRRI, he looked at me. I won't name him, very famous rice breeder. He looked at me and he said, Susan, QTLs are a figment of your imagination. And you'll notice he was not on the list of authors. But I will note that one of the people who is on this list of authors, he will come back into the story a little later. So note Dave McKill. Okay? So then I'm going to highlight this little region at the bottom of chromosome 9 because part of the story I'm going to tell that has to do with Dave McKill comes back around, and that at the bottom of chromosome 9, that QTL comes back around. Anyway, so we had a lot of cleaning and counting and measuring of roots, and I had a team, or Mike Shampoo had a team of people that were all involved. And this team of people would work day and night to cut we had plastic bags in the PVC pipes. We would cut them at certain depths. We would wash, measure, weigh, and with a caliper measure the width of these roots to look at root characteristics. Meanwhile, this is an extraction of DNA. This was a lot of DNA. You needed a lot of DNA to do Southern analysis. And all of these were P32 labeled blots. So many of you are shaking your heads, so I know you've been around as long as I have. Anyway, this was a tremendous challenge. It was we could see very clearly the scientific potential of our mapping, our QTL analysis, and marker-assisted selection was right on the horizon. But this process was really slow. We had to generate biparental populations. We had to do phenotyping and genotyping, and that was RFLPs. It was really laborious, and it was very costly. The enzymes had to come in through Philippine customs. It was nothing like the work that we do today. And each population segregated for only 2 alleles. So we started making lots of different populations because everybody was sure that their particular combination was the one that would be most important. So many people, so many moving parts, so many expensive reagents, and so little connectivity. So information didn't move that easily because these were pre-internet days. And so data sharing was difficult. Data management was beginnings of Excel files, but data analytics, we didn't have the computational power we have today. So all of this made it into a way I started thinking, this is not going to help breeding. This is taking way too long. It's way too expensive. And we're not making the kind of progress. Meanwhile, the breeders were going along independently, and we were completely irrelevant. I disagreed with my boss that these were figments of our imaginations, but we were clearly not on the breeding pipeline. Anyway, I came back to Cornell after 5 years at IRRI, and genomics was moving down the road. We were moving out of RFLPs and moving into SSRs. And one of my grand visions was to be able to genotype the gene bank at IRRI. I thought, wouldn't that be wonderful? If we could just genotype the gene bank, we could make digital selections as to which kinds of materials we wanted to use. And it wasn't exactly as easy as it is today, but we did get our first NSF grant to build a roadmap of natural variation in rice based on, obviously, a selection of the materials in the gene bank. And we asked some questions. How much diversity is there in sativa? How is it organized? And where is it found? And you've already seen the maps and the distributions of ecologies. So now we wanted to add a molecular genotypic layer to overlay on what we thought we knew based on phenotype and adaptation. And what can we learn about genotype-phenotype associations? Can we start making predictions? And what knowledge and tools do we need to accelerate breeding and the management of new rice varieties, given that RFLPs were not going to do the job? So the first thing we did was to collect a sample of diversity from the gene bank based on mostly geography. And we focused on land races. We had some elites in there, but a lot of land races because that's where the maximum amount of variation was, after all. So our collection was from across the world. We were using SSRs, and then we moved quickly into SNPs. And I think our lab here at Cornell has been in the vanguard of providing markers to everyone in the world. We've never protected them. We've put everything into the public domain. And even today, there isn't a QTL paper that's published that isn't using the markers out of the Cornell lab. So I've been very proud, actually, when I go around the world, have so many people tell me that they're so grateful that they were able to use this, this technology as it came out. So what is the first thing we did? Of course, we looked at the subpopulation structure of sativa, and it's well known that there are two varietal groups, the japonica and the indica. And the picture in the upper right-hand corner shows you what my breeding colleagues taught me when I first went to IRRI. They said there are three subpopulations, the javanica, the japonica, and the indica. And the javanica is simply the tropical version of temperate japonica. So those two are the japonicas, and then there was this thing called indica. And they told me everybody in the world who produces high yielding rice is producing indica rice. Okay, what do I know? Fine. We did some analysis using first of all, our SSRs, and we saw sort of the same thing. You know, we saw the japonica group and we saw the indica group, but didn't quite conform to the picture that they had painted for me. And you can see that the tropical japonica and the temperate japonica, the blue and the green here on the left, are very closely related in the PCA. And in fact, they are very distinct in the minds of the IRRI breeders because they grow in such different environments. But they're very closely related evolutionarily. Meanwhile, this huge group that they said was indica turns out to have two very distinctive, evolutionarily distinctive subgroups. But they're not well differentiated either in terms of their geography, where they grow, nor in terms of their morphology, and they're easily crossable. So one is called indica and the other is called Aush, which is a Bengali word for the season of the year in which these rices are grown. Now the Aush subpopulation, as it turns out, had always been considered part of indica. But from this PCA, I think you can see that evolutionarily, it is almost as different from indica as the tropical japonica is from indica. So this was a discovery. It flew in the face of all that was known about rice genetics. And furthermore, we applied the molecular markers to the kind of material that was being grown in the US, which is high yielding and subtropical. And everybody called it indica. And we showed that it's all tropical japonica. Everything grown in the US is tropical japonica. There's no indicas grown in the US. So that was revolutionary. And then on top of it all, I don't know if you see this little thing called aromatic in the middle, that little purple, that cluster of dots. Those are the famous basmatis, the supreme quality rice, which is exported, which everybody, or most people in the world really value. And it showed that actually the aromatic is not indica. It's always called indica because it has long sling grains, and it's not indica and it's not japonica. In fact, it's an admixture between Aush and japonica. And that was even more heretical for the Indians who prized this rice and the Pakistanis who prized this rice as being of indica origin. And it's just been an interesting road for me because I had no preconception about any of this and I have no national identity bound up in it. But things didn't sort out the way that we thought they did. And it helped explain why it was so difficult to make basmati by indica crosses. Because actually basmati is more japonica-like than it is indica-like. Okay, enough of that. So here's some geographical distribution, and I just want to say, if you're step back, like I hope you can see this slide. There's a lot of red and yellow in the top right corner in the Asian, on the Asian continent and subcontinents. And around much of the rest of the world, in Africa and both North and South America, it's mostly light blue. The light blue is the tropical japonica, and the red and the yellow are the Aush and the indica. And so you'll see that the Aush and the indica, which are predominantly grown in the tropical zones there in Asia, are associated with this the high yielding types that we're seeing. But what is interesting here is that it's the tropical japonicas that are grown throughout most of the rest of the world, some of which are very, very high yielding, as we see in the United States and in parts of South America. And so it scrambled people's understanding of both what we could do with our germplasm and how we were combining it. And to some extent, it helped challenge some thinking. If there is a distinct population, which you see as these bright yellow circles centered in the Bangladeshi river delta and in East India, if that is a distinctive, evolutionarily distinctive type of rice, what is it that makes it so special, or isn't it special? Not many people claim to grow Aush varieties today, But part of the rest of the story that I'm going to tell you is what we've been learning as we start looking for stress-tolerant genes, almost 100% of which are coming from the Ausch subpopulation today. So we've constructed diversity panels, and our diversity panels, some of which we constructed here with NSF funding in collaboration with our colleagues in Arkansas at the USDA, and some of which we constructed at IRRI. They consist of all 5 subpopulations, the indica, the Ausch, the tropical japonica, temperate japonica, and aromatic. And these varieties, they are mostly traditional varieties. They've all been purified for at least 2 generations, and then purified from single panicles for 2 generations. And we now have inbred sources of seed that can be distributed. And my lab here, thanks to NSF funding, put 700,000 SNPs on all 1,600 of these lines. And these are now available as the basis for association mapping in rice. The IRRI group then, in collaboration with China, has now added an additional 3,000, and those were fully sequenced. So we have collectively about 4,500 lines of rice, which have been purified, are publicly available, and have genotypes. So the beg for the rest of the community is, what do you want to do about phenotyping? You can phenotype these lines, you can request them, you could request subsets, you get for free, you get your genotypes over the internet. What kind of phenotypes do we want to look at? And we've looked at all kinds of phenotypes, and it's a very large collective now of people who can use these lines or subsets of these lines to phenotype whatever they want in the field, in the growth chamber, in any country in the world that allows them to import those rices. And my lab has been doing quite a bit on panicle structure, on seed size, shape, and color, on stresses related to water, on things like awns. This shows the barbed awn. You may have followed some of that work. On disease resistance, on canopy temperature, and then on this fast growth in response to rising water. Lots and lots of different phenotypes. And so the beauty of this is that the students who used to come into my lab could choose a phenotype of interest, and they had material at their fingertips to work with. And then there's the roots. So the roots are still some of my favorites, and one of the things we did was 15 years after our first root phenotyping experience in the Philippines was to work with Randy Clark, who was part of Leon Cochin's lab at the USDA, and Janelle Jung, who was a PhD student in my lab. Randy built this cool little, this was just the early version of it. It's even cooler now, but this automated or semi-automated phenotyping system where we grew our plants. I think you can see the, grew these plants in a gelum gum. So that's like a sort of clear gelum gum. And then we would rotate them. We put the cylinder, kind of a big cylinder actually, in water. And then we had, that was on a revolving table. And then we had the camera and could take revolving pictures of this so that we could reconstruct 3D images from these 2D captured images. And so this was the early days of what we're calling today high throughput phenotyping and image capture and then automated image processing. And what I just want to say is we would take 40 images per plant as it turned on its base. They were imaged over different days because it's completely non-destructive. And then the software that Randy built, the Root Reader 3D software, could reconstruct single 3D images of each individual's root system. And then, because you could have replicated, these were all inbred lines, and you had replicated versions of this, you could start to look at population level genome-wide associations using these reconstructed 3D images. And this was really quite a powerful technique. And when I think back to the days at IRRI where we were 40 people using a caliper to measure roots, and we couldn't do anything that was non-destructive. This was completely non-destructive. What we were able to do was the following. So this is just one example. We were looking at root system architecture. We used 373 individuals from all the different subpopulations. We measured 13 different traits over 3 days in the 4 subpopulations. We didn't use aromatic. We were not using the basmatis. We were using just the indica, the Ausch, the temperate, and the tropical japonica. Now, the 12 chromosomes of rice are here, And the big lines are the QTLs because we had biparental crosses as well as our association mapping panel. And the little tiny red ones are the GWAS QTLs or the GWAS signals. And what you can see, in some cases, the GWAS signals overlap with the QTL. In other cases, they sort of spanned it. And in other cases, they were completely independent. Of course, the biparental cross was IR64 biasucena, captured only 2 alleles, whereas the association mapping panel captured many alleles. So you can appreciate that they weren't all entirely overlapping. But really, one of the take-homes of this slide is that the QTL resolution was so much lower than the GWAS resolution. And so for the same population size, you would get much better resolved regions of interest if you looked, if you used the GWAS as compared to the QTL or the biparental QTL. So here's a Manhattan plot. And what you see along, I think most of you probably know how to read a Manhattan plot, but these are the 12 chromosomes. And there were 700,000 SNPs, and they're all lined up in order, linear order, as they are arrayed across the chromosomes. And this is the negative log probability. So this is just essentially a kind of p-value for an association between the SNP and the phenotype. And here is the most significant SNP, up at 10 to the -7 on chromosome 3, corresponds to this region on chromosome 3. It was a GWAS hit. And it's an association for root length in the indica subpopulation. And when you look at what these beautiful concatenated images look like, now don't get upset. There is no difference here. Okay. I'm showing you where there is no difference. And it's the SNP that I'm showing you is 66 kb away from the peak SNP. But the point here is these are concatenated images. This one has 39 different individuals replicated to form a concatenated image of what the root length was in these indicas. These are the indicas that carried the A allele at this SNP, and the indicas that carried the B allele at the same SNP. There were 118 of them, and the 2 root lengths are roughly the same. But when you move over and you look at the peak SNP, which is the SNP that was significantly associated with a difference in the phenotype, in the root length, what you see is that the A allele carries the short root length, and the B allele has a longer root length. And there's a slightly different number because, of course, there were recombinants between this SNP and this SNP. So the number of plants in each group has shifted slightly. Now, 2 take-homes here, too. The resolution is about 60 kb. Actually, if you look at the ones in between, you start to see a difference. And so I'm just showing you, you can see little remnants of the difference, but it's not as significant, and the p-value is not as high. So you have a pretty good resolution. In rice, if you have a 60 kb resolution, you're still talking about several genes, and really you have 60 kb on either side. So you have roughly 100, 120 kb. You have at least 20 genes in that region, but you've narrowed your search space considerably. Now, meanwhile, okay, we were doing this work here, our NSF funding. We were very excited about the new phenotyping possibilities. We were moving quickly to get a much higher resolution view of the associations between genotype and phenotype. And our colleagues at IRRI had continued down the road of the biparental mapping for 15 years. And they were doing amazing work, measuring, not only with calipers, but they'd also started to automate their phenotyping. And they were able to do it in the field, which I clearly was not. And so some of the collaborations that I've had over these years have kept me in touch with this work. And I just want to say that while I was doing the work here, they were doing the work there. And one day at PAG, the 2 groups were getting up. I got up to give a talk, and my colleague at IRRI got to give up a talk right after me. And we realized we were doing the same work. He was doing it in the field, and we were doing it with this automated phenotyping. The question was, are we discovering any of the same QTL regions? And I just want to show you that this work involved planting rice seedlings into a spaghetti colander, which was then planted into a tub, which was sunk into the soil so that you could manage drought, if that's what you wanted to do, and so that you could pull it up and clean off the roots, much as we had been doing 15 years before at IRRI. And what you see is then you could clean off the roots and then clip the roots, and you could measure them as they came out of the colander. And so the different phenotype that was being measured by this group was whether the roots grew straight down or whether they seemed to sprawl out across the soil at a different angle. And another group at IRRI had been doing phenomenal work looking at what we call phosphorus uptake. It's called phosphorus starvation tolerance. And again, they had come back to the understanding that it was a root phenotype that determined phosphorus uptake and scavenging ability. So back to my theme about roots. And the next slide shows what the discovery that was being made by the groups who had been doing this for 15 years in the field entailed. And it entailed a cross between IR64 and a traditional upland variety from the Philippines called Kinandang Patong. And Kinandang Patong had very deep roots because it was an upland variety. And IR64, which is the quintessential indica out of IRRI, it is planted on more acreage even today than any other rice variety in the world. And it has very shallow roots because it's grown in the irrigated system and it doesn't need deep roots. And he crossed those two, and then he back-crossed and introgressed the region with the largest effect QTL for deep roots into the IR64 background. And he created a near isogenic line of IR64 carrying one region on chromosome 9 that carried the deep-rooted QTL from Kinandang Patong. And the trait donor was, of course, one of those early landraces that we had selected for one of those biparental crosses made back in the early 90s, and that was publicly available. And what did he discover when he cloned that gene? He found that this gene, which he calls DRAW1, and many of you are familiar with this work, that a single base pair deletion in exon 4 changed the root angle, and that change in root angle, so this is the deep-rooted, and this is the shallow-rooted, and that change in root angle had no effect in yield under irrigated conditions. But when under moderate drought, the IR64 could no longer keep up with yield, and the NIL that had that one introgression on chromosome 9 associated with that QTL was doing quite well. And under extreme drought where IR64 died, the NIL was still able to survive and produce offspring. And so what is really remarkable is he showed that that one base pair deletion in exon 4 that changed the root angle enhanced grain yield under drought. And he could show it because he developed the NILs. These are pairs of lines that can go out and be planted in the field under the same conditions. Now, for those of you who are wondering what this gene does, it's an unknown protein. It has no correspondence to anything with known function. But we do know that it's negatively regulated by auxin. It shows a higher expression of the DRAW1 protein causes the downward root growth. So it is quantitative, and there are variants across in the germplasm array that have more or less production of the DRAW1 protein. It's involved in cell elongation at the root tip, and it localizes to the plasma membrane. And I'm going to move quickly because I have a feeling I'm running out of time. I want to go on and say, and I hope Kenong Yu is on in Geneva, because I'm going to feature some of the work that Kenong Yu did he did it originally at UC Davis, but this is an important story, and I really want to tell it. It has to do with the sub1A gene, which confers submergence tolerance. So the one I just talked about was DRAW1, which is related to the downward growth of roots, which confers drought tolerance. And this is a different phenotype entirely. It has nothing to do with roots, but this is a phenotype now that is of a great importance in the same rain-fed systems where the drought is important, and this is for submergence tolerance. And in this case, the submergence tolerance is conferred by what they call the sub1A gene, and it's not a single base pair change in a gene of unknown function. It's a complete addition of a new member of an ethylene response factor gene family. And so in this case, Kenong showed in his work when he actually cloned this gene that the sub1A was the new member. The B and C are shared with the reference genome, but the A gene was not present at all in the reference genome, which is why it was a challenge to clone in 2006. And this particular donor was called FR13A. It's an Ausch landrace. It carries this incredible opportunity to survive under submergence, and it basically holds its breath. So it's an ethylene response factor gene, and I want to tell this bit of the story. Because if you look at the citation that's up there, some of you may recognize the name Kaufman, William Ronnie Kaufman. So Ronnie Kaufman was a breeder before myself and before Dave McKill. He was at IRRI in the 1970s. And one of the papers that he produced in 1981 was called Inheritance of Submergence Tolerance in Rice. And the take home of that paper was that there was a single major gene associated with submergence tolerance, and that it would be important in the rain-fed lowland areas throughout South Asia where flash floods were a problem. That was followed by a paper by Dave McKill, who came in as Ronnie's replacement for the rain-fed lowland breeding program at IRRI, and he published a paper where he had actually improved semi-dwarf varieties, which were high-yielding varieties, by introgressing this gene into those varieties. And Dave McKill, when Ronnie came back to Cornell, Dave McKill took his place at IRRI and was still there when I went there. And Dave McKill was trying to breed based on phenotype, and he had to flood these things and see which ones survived, and it took quite a long time. And so when I got there, Dave McKill came to me and said, I know exactly what I want to do. I want to make this population with this donor because it survives under submergence, and I want to do a QTL analysis with your markers, and I want you to give me markers so I as a breeder can start moving it into the backgrounds that I really want. And so that project, which again started in the early 90s, took the form of eventually a cloning project back at the University of California. And Kenong Zhu, who is today an apple breeder up in Geneva, did this incredible work where he had to clone a gene that didn't exist in the reference genome. And it was quite challenging for him. But once they'd cloned it, they were able to move this gene quite readily into other backgrounds. But let me just say, before they'd cloned the genes, they knew where it was based on the early work, and they had markers. And those markers went to the genome mapping lab that I had initiated at IRRI, and people started putting it into the major varieties that were planted in the flooded zones across South and Southeast Asia. And so they had a line in the Swarna background, which is one of the most commonly planted farmer-preferred varieties throughout this rain-fed lowland area in India and Bangladesh. They had put the sub1 gene through backcrossing, had put it into Swarna. And those varieties were already in the early testing phases. And what I'm showing you here is what happens under under submergence of 10 days. So 30 days after planting, the crop is beautiful, and then a flash flood comes and sits on top of the rice for 10 days, and it pretty much wipes out the Swarna that doesn't have the sub1 in it. The sub1 has come from an Ausch landrace called FR13A, and when you have that introgression in your line, your material holds its breath until the water subsides. It can do that for about 14 days, and then when the water subsides, it can regrow. And this saves people from what they're doing here is they're trying to go back in and replant that field in order to salvage their crop for that season, where here they don't have to replant. So that is the value of this gene. And it took a lot of foresight because the first crosses were made in the early 90s, and the gene was cloned in 2006. And then the back crossing had already begun at IRRI once they had markers from the earlier biparental QTL mapping. And what we're showing here is the number of different backgrounds that they used for introgression of that gene, as Dave McKill, with great foresight, thought, if we're successful, this gene needs to be put into different backgrounds for different countries where different farmers have different grain quality preferences. And if we get it into the varieties that they already like, then it'll be an easy swap for them. We're not asking them to do anything different than they've already done. We'll make near isogenic lines, and they'll see for themselves whether this is better. And so this started at IRRI, and this was Septi's work. Septi also was a student out of my lab who then was at IRRI doing this work. And they moved it through backcrossing into a variety of different backgrounds. And what you see in this graph is that the sub1 varieties always outperform the non-sub1 varieties. And under increasing days of submergence, although they all decline a bit, the submergence quality, the sub1 varieties always do better. And so Within 5 years, the first releases, the actual first variety releases were in 2009. Within 5 years, 10 million farmers and 10 million hectares of rain-fed lowland land, of flash flood-prone rain-fed lowland land, were covered with sub-1 varieties. So the backgrounds are different. This one gene is the same. And there were 450 different government, NGO, farmer organization, and seed companies that were involved in getting this material out from IRRI and from that breeding program out to farmers. And that is one of the record uptakes. And I think the fact that they chose to do this by not redefining the variety, just redefining one trait, helped with that uptake. And I just want to say that they had a whole strategy for doing this. And being part of a strategy, any of you who do work, that even if you start by cloning a gene, if you can plug into one of these networks, your work may take off and really have an impact. And it's very important that you understand the strategy behind the work that you do, because these strategies are not simple. They involve many, many, many people, public sector, private sector, government, NGO, as we were saying. And in this case, here are a few pointers. The sub-1 gene was introgressed into farmer preferred varieties, as I've said, each one different for a different target site. The flood-prone areas themselves that were being targeted were identified using remote sensing, GIS, and ground intelligence. In other words, there are a whole slew of people in the government already using digital technologies to figure out where the most likely occurrence of certain stresses is, especially with climate change. At-risk villages then were targeted for variety that we call participatory variety selection, or PVS. And this was all prior to release, so it was part of the selection process. And they used male and female farmer input to make sure that women who were on the side of preparing this on the culinary side, or who were involved with the transplanting, were involved in the decision making. They also included local checks so everybody could see for themselves whether these varieties were something they wanted or not. And instead of trying to plant one target area with a large expanse, they chose to distribute mini kits. These are about 5 kgs of seed per village. They were provided to thousands of targeted villages to allow for farmer-to-farmer dissemination of seed. The seed is all inbred. Farmers save their own seed, and when it's good, it moves very quickly from farmer to farmer. So they didn't require seed companies in the beginning. And the coordination that was required was quite considerable, and this was all coordinated under what they call the Strasser Project, thanks to the Bill and Melinda Gates Foundation. And the Bill and Melinda Gates Foundation is very big right now in trying to provide the kind of coordination that is needed between the scientists, the breeders, the government agencies, and the technology exchange that is required to really make this kind of thing work. So I'm coming down to the end of my talk, and I just want to say there's a lot of beautiful stories about the one gene at a time that make a big difference. And sub-1 and the semi-dwarf before it, and the pistol, which is the phosphorus starvation tolerance gene, and others. I could tell many of these stories, but really the question is one gene at a time enough? We have to think about what's happening around us, and the climate is changing faster, and people are fleeing the countryside in droves, partly due to climate change and the civil unrest and the disasters that follow. Particularly, as I go back to think about the beginning of the talk, because the export market for rice is so inelastic that when there's a crop failure due to climate change, there isn't enough in the international market to make the rice available that people need. So there's a massive kind of feeling of unrest and an accelerated desire to move breeding forward. So I know I've gone a little bit over time, and I apologize. I just want to take you down this road. So we have the rise of the Walmart model. We're all affected by it. And breeding is affected by it as well. You generate infinite amounts of data on every aspect of your subject matter. Maybe it's toothpaste, right? Walmart, it's not all about breeding and other people that need our products. You use machine learning algorithms to define patterns and generate hypotheses. In other words, you learn from the data. You validate your models, you redefine your hypotheses by testing on new populations and new environments. You know, the way that Walmart moves is they figure out what works in one population. They look for the population that's most like it, and they take that exact retail model and they start applying it somewhere else. And it works really well. You use validated models to predict behavior or performance of future populations and environments. And you beat the competition using data and intelligence to speed up the delivery of products that people want and sometimes that they need. This model is the model right now, the big data model that most of us are working under, and it's a challenge. We have our parallel inbreeding and genetics where you collect lots of data on phenotype, on environment, on genotype. You develop models as quickly as you can that integrate all of those and help you predict the performance of your lines. You continuously update your models, you iteratively improve them. You use predicted breeding values to increase selection intensity, increase accuracy and decrease cycle time, and then you modify your breeding design to take advantage of new efficiencies. That's the name of the game. It's the Walmart model in breeding. And we're all being asked to try to accelerate for reasons that we all understand. But it's taking us down this road of more, constantly more, faster, and hopefully better. But it's creating this two-world phenomenon, the thing I call, or my colleagues over in engineering call, the digital twin phenomenon. You have breeding pipelines, which are basically a collage of genetic resources, breeding populations, and improved lines that are crossed, evaluated, selected, and recombined in a recurrent cycle in biological time under field conditions. That material is physical. Knowledge about that material is dynamic and evolutionary. And side by side, you have a genomics pipeline, which is a collage of algorithms and analytical procedures that process, reprocess, intersect, and integrate data in parallel computing streams, generating a series of models and predictions about future performance to guide selection. The information is representational and it resides in cyberspace. And hopefully it reflects the reality, the physical reality of the food that we need to produce in order to maintain livelihoods and to maintain adequate human health. But the challenge for our generation, or for actually I would say the younger generation, is really the challenge of how you're going to integrate automation and big data into a conception of the world that still maintains touch with reality. We have digital tools, data streams, simulation, and prediction, and it's completely revolutionized the way we see the world and our ability to make predictions about the future world. It's imposing disruptive change. And my question as I come to the close of this is, can we integrate the best of both worlds? Can we continue to discover diverse alleles and mechanisms that confer resilience and nutrient use efficiency, and also to use genomic selection or genomic prediction to efficiently combine them with all the alleles that we have for yield and quality in our elite breeding material? And I want to just end by these few thoughts I have at the end of this presentation, which is that beyond breeding and statistics, rice is also a bridge to human history and deeply held cultural values and perspectives. It is a source of beauty. It's an essential natural resource. It's a product that provides livelihoods, promotes health and well-being, and it's the foundation for partnership, education, and innovation, and for me, a source of inspiration. So with that, I thank you all for listening, and I thank the group that I've worked with and my funding sources over many years. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.